Hello friends, today we are going to discuss a very important topic which is just next to my previous video after the DNA chemistry. I want to start this replication part. So basically the eukaryotes has a linear DNA, and as I have discussed today, I am going to restrict my entire discussion on prokaryotes or prokaryotic replication. So in the DNA constitution, if you see, the prokaryotes are a very simple organization and they are a circular double-stranded nucleotide. It is double-stranded circular DNA, and as you know, this is eukaryotic, and our eukaryotic linear DNA is also double-stranded but has two ends. These ends actually we call telomere. So if you think from the enzymatic involvement, so prokaryotic replication differs in a very enzymatic way, because if you consider the enzymes that are involved in prokaryotic and eukaryotic replication, they are also very much different. Say if you consider the enzymes, the telomere part, which is not present in this diagram because it is circular. So bacteria or any prokaryotes do not have such end problem, linear DNA's end problem. So in case of the prokaryotes, you will not get any enzyme to replicate their ends, but in case of the telomeres, in case of the eukaryotes, we have telomeres. Yes, telomeres, an enzyme. RNA dependent DNA polymerase, an enzyme which is specially present in eukaryotes. So it is not present in our prokaryotes. But what I want to emphasize that from the DNA nucleotides level, the basic construction process is same. Both of these are formed from the A double bond T or G triple bond C. So these bonds are present here also, and this nucleotide within this nucleotide. So there is no basic difference in case of the constitution of DNA in both cases of circular DNA or linear DNA. But the prokaryotes has to some extent similarity with the eukaryotes. Say if you consider these eukaryotes, they have this DNA, linear DNA, and they have the circular DNA. But in case of eukaryotes, you will see many origin of replication is formed. and in case of the eukaryotes, you will find a single origin of replication. Single origin of replication. Single origin of replication. So this single origin of replication, and here is linear and multi-ori or origin of replication. Origin of replication. So now the question how this structure will get, basically this bubble and this origin bubble in case of many origin bubble in case of eukaryotes. So the basic thing is same up to this. The bonds need to be melt and DNA should be relaxed, because you know sometimes DNA remain in a supercoiling form. Supercoiling form. So it may be negatively supercoiled or positively supercoiled. So basically because it has the negative and supercoiling in here also. So to start this process, you first have to melt these bonds. So which enzyme will help you? This is the question. So first, enzyme called DNA helicase. Helicase will act on this. Here also the helicase, prokaryotic helicase will work and it will melt down the nucleotides in and around the starting point. So I am not going into details why this place is selected, or you can say that as the number of the double bonds are reached, the site is preferable, because you know the less the double bond, the less the energy is required. So helicase will melt down this helicase will melt this hydrogen bonds and it will use ATP hydrolysis. So ATP hydrolysis, there is the ATP hydrolysis will help to create the bubble. So first, the supercoiling is DNA supercoiling, which is a problem to this linear or especially the E. coli bacteria. So the bacteria will use some topoisomerases. Topoisomerase. So here it is 1, it is 2. Please notice this number, because one by one the enzyme will come into play. So this helicase second, but first the coiling is removed so that DNA can be found in an extended conformation. So after this, the supercoiling is reduced by topoisomerase 2 in case of the bacteria. The double stranded break is made and this transient break will create the access to other enzymes. So now helicase, up to this the helicase is in existence. After that the thing is different. So from here we will start the only discussion of our eukaryotic prokaryotic bacterial DNA. So helicase and topoisomerase already in existence. So now you have got your activity of topoisomerase 2 to relax the DNA by cutting the double strand or creating the double stranded break. It will relax, it will form bubble, and then helicase into existence. helicase will melt the melt the hydrogen bonds and bind and stabilize the DNA in a single stranded conformation. So binding after this, the helicase after melting will take place and it will remain within the whole process. So now you will get, from suppose you take this side, you will get such a conformation. Say you get such a conformation. So if 5 prime to 3 prime and 3 prime to 5 prime. Already I have discussed how this 5 prime and 3 prime is in the convention, because it is only during the polymerization phosphodiester bond creation we get the direction. This is the direction we are discussing about. And let me concentrate on this enzyme. So now this melted portion, which just like a bike and it's a fork we call, and here we have one DNA, this term 5' to 3', it is we call template. This template always want to reanimate, or it wants to coil up on things. So in such case, the third player is SSBP, that is single stranded binding protein. So single stranded binding protein gives the unwinded DNA strand in an extended form, that is it maintain the single stranded form for replication. So it is very much essential to maintain or prevent from formation of the bonds. So this is done by a special protein, we call it single stranded binding protein. So it will prevent from reannihilation. But now you need to synthesize this strand. So our fourth enzyme is our fourth requirement is a formation of a primer. So it is called RNA primer. It is generally we get from an enzyme called primase. So primase are basically RNA polymerase. So it will form this strand, and it is complementary to the DNA strand, and this primase, which is synthesized by RNA, or the primase, which is basically an RNA polymerase, which synthesizes the primer, it is approximately 4 to 10 nucleotides long, and it will form a hybridized DNA RNA, and it will have a 3' free hydroxyl end. So as from our discussion in the previous video, I have discussed how this polymerization reaction takes place with the help of this 3' hydroxyl group and with the NTPs, which is very much available in the growth medium or within its cytoplasm. So The D entities nucleotides, suppose it is D, A, T, P, D, and then guanosine triphosphate, D cytidine triphosphate. So it will, the nucleotide will one by one move and it will extend the chain. But this extension has its own story, because in case of 5' to 3' chain, you will get a continuous synthesis of the new strand. Continuous synthesis. So this continuous synthesis or continuously nucleotide is added. So this continuously synthesized template strand is called a leading strand. Leading strand. So this leading strand, now with the help of primer, with the help of this primer, RNA primer, it get access of this nucleotides from the group medium or cytoplasm, it one by one added. But this process is dependent on an enzyme called DNA polymerase 3. So our third and fourth enzyme in play, because we have fourth enzyme as a primer or primase, RNA primase, and fifth is DNA polymerase 3. So DNA polymerase 3. Now it is continuously synthesized and you will get a complementary one single strand. But in case of the 3' to 5', here you see the direction, orientation is reverse, because here you have 3' and the polymerization can be expressed very easily, because it allows, the orientation allows the polymerization. But in case of this strand 3' to 5', you need to the bacteria need to evolve some tricky mechanism. So what did this bacteria and our cells also, we'll discuss that later. It has synthesized primer, but apart from the fork, if you consider this is the fork mouth, it is moving out, and these are the small, these are very small RNA primer. The same enzymes are involved, but in this strand 3' to 5', they have many primers, RNA primers. So now they form multiple RNA primers, and these RNA primers, now with the help of DNA polymerase 3, already I have discussed, it will form small synthesis. This small synthesized segment of DNA, it is around 100 nucleotide base long, 100 nucleotide base long, and it forms short fragments. So these fragments are called on this strand Okazaki fragment. So now at new term you get, this is Okazaki fragment. This red strand is, the previous strand is Premier strand, which is called lagging strand, and the synthesized, newly synthesized, this short strand, these are called Okazaki fragments. Okazaki fragments. So Okazaki fragments. Now you have these Okazaki fragments. So up to this, you will have strands, leading strand and lagging strand. They are somehow have synthesized the Okazaki fragments and continuous synthesis of the replicated content template. From template it synthesized. So now you see there is some problem. RNA primer become a problem to this lagging strand as well as to lagging strand and as well as leading strand. So you need to eliminate this fragments of RNA primer. So in this case, another enzyme will come into play, and that enzyme is DNA Pol 1. It's a very interesting enzyme. DNA polymerase 1. It has 5' to 3' exonuclease activity. So far my knowledge is concerned, this exonuclease activity means it will cut from 5' end and it will eliminate all those 10 nucleotides. These are the which are the part of RNA primer. So you will get DNA strands which are discontinuously synthesized, discontinuously synthesized within the lagging strand, excluding or with the help of this DNA polymerase 1's 5' exonuclease activity, it will eliminate out. So now you have this conformation, and now you have to synthesize this thing. So how these gaps, these are the gaps, how these gaps are synthesized. So DNA polymerase, which has 5' to 3' polymerase activity 2. So it will synthesize the nucleotides until the next discontinuous Okazaki fragment is made. But when before that, the final sealing is need to be done. So how the final sealing will be made? With the help of DNA ligase. So DNA ligase will help to seal the gap. So until you use all the enzymes, the replication is a very much problematic one. So bacteria has done all the involved all the enzymes, but always the helicase remain active, because if the helicase do not remain active. So along the movement it need to be proceed forward. So during this forward movement the helicase will release the nucleotides from this coil of DNA, and here topoisomer will also work. So once again I want to recapitulate the things. So the recapitulation part or the whole thing. So first you have used topoisomerase. Then you brought into helicase and it will remain in an active form, and then you have SSBP, single stranded binding protein, which bind and stimulate the DNA in a single stranded conformation after melting by helicase. So now the DNA polymerase 3 will come, and these two DNA primer DNA polymerase 3, but before that you need to synthesize primer. So RNA primer, it is by the help of primase enzyme that will help to form. So the primer, which has a 3' OH immediate conformation found within its ribose 3' OH. Yes. So it will extend the chain in two fashion with the help of DNA polymerase 3 as DNA polymerase 3. So DNA polymerase 3, which has 5' to 3' polymerization activity, and this has also 5' 3' to 5' proofreading activity. Means if any misincorporation, whatever the miss is in the template strand, if it deviates, then this proofreading might help. So today I am not going to discuss that very much. So this 5' to 3' polymerization reaction will help to form the continuous chain and discontinuous chains. Then the removal of primer is required. So removal of primer is done by DNA pol 1 by the help of 5' to 3' exonuclease activity. Exonuclease activity. So it will help to eliminate from these parts, and there is 5' to 3' polymerization also. So this will polymerize up to certain point, and then the final resealing is required. So this sealing is done by DNA ligase. So within this